Hi everyone and welcome to this lecture. So here we have the application, the project in which we will apply the algorithm of clonal selection. So let's get started. So what is this project about? This is actually a project of digit recognition or generation. The objective is to recognize the digit in the right, represented by pixels, to generate this digit right here and therefore recognize it by pixels, and to see how random patterns through a process similar to that of evolution can generate well-defined patterns. The figure with the number 8 in pixels, here's the problem antigen, is the objective, is represented by a vector or list with several values of zeros and ones, just like the antigens, as we will see later. So this is how we can represent the antigens and the antibodies. A vector or a list with a lot of zeros and ones. And we can turn this into a two-dimensional vector into an image of pixels. So here we have a random antibody. So here we have a non-matured antibody. Initially, the antibodies look like this. However, the objective is to look like this, to match, to recognize the antigen right here. So this is the antigen. The antibody must recognize the antigen. So here we have a process of iterations where in each generation the antibodies are matured. So here we have a process in which the populations of antibodies are maturated over each iteration, over each generation. So here we have a completely random antibody at the beginning, and here we have the antibody being matured through the iterations, through the generations, and finally we have the completely matured antibody which matches the structure, which matches the image of that of the antigen. So this is our objective, and this is what we will see when we run the algorithm of clonal selection. So how do we build the objective functions? As I explained previously, both the antigens and the antibodies will be comprised of vectors formed by zeros and ones. We have to build a function that iterates over every position of the antibody and the antigen. So we will compare, for example, the first position of both the antibody and the antigen. In this case, the score is 0 because they have different colors. In this case, the score is also 0. In this case, the score is 1 because they have the same color. So we update the score of affinity. So this will be the objective function of the problem. We have to remember that the maximum affinity value is 121 because we have 11 elements here, 11 pixels here, and 11 pixels in this dimension. We have 11 in height and 11 in width. So this is why the maximum affinity value is 121. And this was the lecture where we introduced the problem in which we apply this algorithm.